So it's a great pleasure to be in beautiful Mondello and participate in this HBP school. I appreciate the invitation. Neuron has been associated with the Human Brain Project from the beginning, and before then with the Blue Brain Project since 2005, when it became began to receive its parallel abilities, basically through a collaboration with the Blue Brain Project. And over those years, it's developed a great many parallel idioms having to do with load balancing, a lot of the practical implication aspects of parallel computation, and especially parallel setup, and also effective use of supercomputers, high-performance computing. Prior to that, Neuron has also been supported, long-term support, through the National Institutes in the United States of the National Institute of Neurological Disorder and Stroke. And in fact, now it is in year 42 of that, of a project that has been fairly continuous over that time. And at least until year 46, has just received new funding, I think in large part based on the parallel work that's been done with the Human Brain Project and the Blue Brain Project. So over those years, it's developed into a huge bag of idioms and devices and elements that are of practical use for developing simulations of neurons and neural networks. It would really be impossible to go into any detail in this short time, so I'm just going to kind of show and demonstrate a few square inches of the fabric and give you kind of an overall idea of how it goes about its work and how one develops a simulation and what Neuron is about. So classically, Neuron has always excelled in its ability to simulate or do simulations of neural systems in which, in your judgment, cable properties are important in the sense that it would be too much of a simplification, again, in your judgment, to reduce these complex cells down to single compartments. I mean, that can be done, but when it's done, it probably ought to be compared, at least in small systems, with the results that one obtains when cable properties are necessary. In addition, from the beginning, it was also designed with the idea of fairly complex membrane properties in mind. That means any kind of sodium and potassium channel, or pumps, or calcium diffusion internally, or reaction diffusion inside of the cytoplasm, and transmitter release and things like that, sodium-calcium exchange. In addition, it is good for single-channel simulations where there are local extracellular fields adjacent to the membrane. Linear circuits are very important in a lot of cases in order to simulate the instrumentation, which involves stimulating and recording from cells. Obviously, networks require synapses and on occasion gap junctions, although you ought to avoid them if you can abstract them away in your model. But they're simulatable. The typical abstraction used for networks of spike-coupled neurons is merely to note that however complicated the dendritic process computations are to generate an action potential, in the end, along the axon, it's a fairly stereotyped response which propagates pretty much independently of how it was generated. And so the way this is abstracted away is one just removes the axon from the model, and instead, at least at some short distance, when the cell has produced a stereotyped response, that at that point, a certain trigger point on that response becomes then abstracted as a discrete event, a logical event, which then gets delivered to the target cells at certain specific delay times, which you can vary those during a simulation, but generally speaking, they're just constant delay to that point, to that point, to that point. So we retain a lot of the complexity or all of the complexity in some cases, and complexity of the membrane, complexity of the dendritic trees in this region, but this just, in that point, becomes just a logical event, which is delivered to target synapses, and that logical event then causes a discontinuity in the conductance or some state of the synapse, and has its effect then on the target cells. So always from the beginning, I think it's really important to always keep in mind the distinction between the physical system and the model, the conceptual model. It's this transformation of abstraction and simplification that requires your scientific judgment. You're the ones who develop a model, or you're the ones who have to know what model is being simulated and be able to criticize it, understand its limitations, what it has left out, what it has kept in, so forth. It's the purpose of simulation, though, to be as faithful as possible on the computer in terms of what the effect or what is the simulation doing should be exactly what the model is doing if it could be actually realized in reality. In terms of these, basically, typically the model ends up as a mathematical set of equations, and we want to faithfully represent these equations. And so in the model, in this case, there's a squid here with an axon, and these are the Hodgkin-Huxley cable equations. And the neuron representation in Python is that we import the neuron module into Python. We create an axon with H dots. We imported H, which is the entire capability of neuron now in Python. And using Python syntax, we say, create a section, which is the cable. The axon is that long cable, and we divide the axon into 75 compartments, and that's the first part where it differs from the model itself, which was a continuous set of partial differential equations. We've divided into compartments, set a diameter and a length, and we've inserted the Hodgkin-Huxley channel. So the section itself is just this part of the equations, cable equation. And inserting HH inserted all of this from here on. And now that raises an entire new set of questions. Well, how faithful is the simulation, the computer representation, relative to the model that you have? So we have a kind of double process of problems. In other words, the model may not very well represent the physical system, or may well represent the physical system. And the simulation, though, should always be a good representation of the model. And I believe over the years, we've developed techniques and so forth in order to be fairly reliable in the solution of these model equations using using the simulator. Now, the overall structure of the program has always been that there was a significant compiled section which does the high-performance computations, and then all the rest, classically there was an interpreter using the Hoke language, which was invented in 1984, which with neuron-specific syntax in which people always generated their models. And there's a great deal over the years of legacy models and code, in this language. Since 2007, the Python interpreter has been available to do all that could be done with these things. So this is the actual number crunching part, and this thing then is the description part, which defines the model. And it's up to you then to define the model, and it's up to the neuron developers to make sure that what model you define is properly carried out on the computer. In addition, because you cannot put in a single thing all of the, a priori, all of the behavior that you need at the high performance level, there's a membrane channel specification, or there's a model description language in which one can add fairly arbitrary code into the program itself, into the part that's being simulated underneath the interpreters. And most channels, for example, are expressed in a fairly descriptive way using that method. Now, the network construction always, in my experience, has ended up as basically a three-part process in which one defines the types of cells that are going to be used in the network. And there's usually a small number of types. By small, oftentimes 2 or 10 or 100. However, we need to then create each cell in the network by instantiating up to thousands or millions of times each of these cell types. The types can be parameterized so that each cell can be unique. For example, a unique morphology, a unique set of membrane property parameters such as how things are located, how things are the values of various parameters. But in the end, this is basically the easy part of the problem. The hard part of the problem in the end is connecting the cells. And so I'll probably spend a good deal of time at the end of the talk in some idioms that are involved in doing that. Just to very briefly mention cell building, the cell building process, or I should say the creation of a cell type, is fairly mature in terms of its our abilities to do that in general. And it's actually been encapsulated in a graphical interface, which you can build a cell type, in which you basically draw the general topology or architecture of the cell in terms of regions, okay? Set those regions into as subsets, and then specify, either specify or not a geometry, but in the biophysics, in each of the regions, for example, in the soma in this case, or the dendritic region, you can specify what membrane channels are present in that region. The only thing remaining after you've defined a cell type like that is to select a location which is the place one will look for the discrete events, the generation of the trigger points for the discrete events as they're generated. And usually it's the end of a short region of axon which is long enough so that the action potential has reached a stereotypical shape instead of something dependent upon the dendritic input, and so forth. And the last aspect then is that you've got obviously sprinkle synapses everywhere on this at proper appropriate points on the cell. That's just a brief description of that. And it's also possible with these cell types then to, for an instantiated cell, apply a 3D reconstruction to the, read a 3D reconstruction file and apply that into the cell builder, okay, result. And this is what's done with all of the Human Brain Project simulations, in which there are several hundred types, several hundred of morphoelectric things. We can combine those with morphologies to generate, you know, obviously a very large number of cells according to desire. Okay. So export to 3D cell build. Okay. So in the network, after we've created all of the cells that we're interested in, then the problem becomes how do we connect them together? And that's done with the network connection object, which essentially you need a presynaptic object and a postsynaptic object, or a synapse object. And in this case, you see the presynaptic object, in this case, is the pre-cell, whatever that object reference is, and the axon at location 1, and we're referencing the voltage, meaning that you know, it's the voltage that we're watching for it to cross a threshold. The other end of the netcon connection object is whatever synapse happens to be that we're connecting to. In this case, for specific purposes, I've expressed it as an exponential synapse, which is built into Neuron. You can modify synaptic properties to be as complicated or simple as you like. The exsyn means that when the event arrives, the conductance instantaneously, discontinuously changes by a weight that's given by the synapse, the netcon weight. And then that conductance decays exponentially with some time constant until, and then another synaptic event, discrete event as it arrives, will then ride on top of that decaying exponential. So that's one of the simplest forms of excitatory conductance changes synaptically. Every netcon can have its own delay. I would say in most models it's set to a constant value of 1 millisecond, but in complex models one measures the distance along the axon to its target, specifies the delay that way. The delay should also be considered to be maybe the synaptic delay as the spike arrives at its target. There's a bit of time for all of the machinery to release the vesicles, for the vesicles to diffuse across the cleft and bind with the postsynaptic channels, and then they begin to open for current to flow through. So you can see what the abstraction is here with the network connection. We can still have a great deal of complexity in this operation, but this is just basically a threshold detection process. So one connects the cells by, creating these netcons with their pre and post, presynaptic and postsynaptic targets, source and targets. The problem with parallel is that these cells may be on different machines or different processes. And so it's not really possible to say when you correct the netcon what the presynaptic voltage is because it doesn't exist on this machine. But we do take the tack of always creating the netcon on the machine where the target exists, because then we can keep this delay, we can keep the weight, and obviously the cell exists there to complete the processing of the model. The way this is done in Neuron is with a object called the parallel context. So you'll see the PC equals h dot parallel context once, and then PC is used hundreds of times then throughout the model code you do. And what it imagines is that every spike source, oftentimes we think of it as a cell, but actually it's a particular location on a cell looking at a particular state variable for threshold detection. It must have a global identifier. I've just for an example, I've said, well, let's suppose this is processor 2 and this is processor 4 on a cluster, and we're going to imagine that on this processor, we're going to associate a global identifier. 7 is going to be on this processor, and global identifier 9 for that cell will be on that processor. And a lot of times, this is part of the conceptual model in which one imagines a global set of cells in the network and identifies each of those cells with an identifier, an integer identifier. And one way of distributing these identifiers, because we haven't created anything yet, we haven't built any cells, we're just distributing identifiers to the various machines, is through the Python range variable in which processor ID, rank 0 of the processor 0, that will contain GID 0. And processor 1 will contain rank 1 will contain ID 1, and so forth. And then we keep going until all in cells are distributed, but on at each point you skip by in host. So if there are five hosts, then rank 1 will have 1, 5, 10, 15, 20. Like a card dealing algorithm. So we can distribute 1 million or 1 billion identifiers among 100,000 machines this way, just the same code, or 50 identifiers among 2 machines. But then you have to associate the global identifier with the cells themselves. So in this case, it was very simple. I've got a vector of GIDs on this rank, and if I have a uniform model in which all my cells are the same, right, then I'll just say, well, let's create a dictionary between GID and the cells for GID and GIDs. In other words, this basically ends up creating all of the cells in the network. But now we have GIDs and cells, but there's still not an association between them. Okay, it's only conceptually through this dictionary so far. So for the GID in cells, I then say to the system, the internal system, that this GID belongs to this processor, and I associate that GID with a particular, not just the cell, but a particular threshold detector. So in this case, it would be h.netcon, cell of GID dot axon of 1 dot, and then the reference to the voltage at this location. Now that that's been assigned, notice that there's no target at this point, and in fact, the netcon is immediately destroyed because there's no reference to it after it returns from the function, the pc.cell function. And so, but that's enough to record the presynaptic location of the that you will always use to connect. And then, basically there's a netcon list, and you append you append to the list a connection, which is PC connect on the target machine. This must be executed on the target machine of a source GID, okay, which does not exist necessarily on this target machine, but on some other machine, and the synapse that you're connecting to. And usually, this is effectively always in networks, essentially a double loop. You loop over all of the target GIDs in your cells list, okay? And you loop over all of the source GIDs and synapses for the connections you want to make. And here is where one can talk in general. Essentially, you've got to make all the connections, but it's the problem of network construction to generate the code that you need in order to make these connections. Okay? But it's not a neuron problem, it's a logical problem from your point of view. What are the sources? What are the targets? You know all of the targets, all of the cells, but how many synapses are, how many netcons, and so forth. And that can generate huge amounts of code. And in fact, it's not unfair to say that there are 3 or 4 large-scale projects ongoing in the world, or well, just having to do even with Neuron, which is how to make these connections. Right? It's a very complex problem. In fact, we don't know the connections of the brain. In fact, so one makes abstractions, decides what kind of connections you like, all to all, neighborhood. What makes a connection is a very complex property, how to define whether a connection is supposed to exist or not. In abstract networks, it's very simple, but in biophysically realistic networks, it's very complex. However, after you do make the connections, all right, because there was always a delay, every netcon has a delay associated with it. It's axonal plus synaptic delay. It's never 0, although in Neuron it can be 0, but then you're restricted to a single process. When there's a delay, there is a minimum delay of all of the netcons. And what that means is that any spike generated in a minimum delay interval, which may be a millisecond, right? Hopefully, maybe a millisecond. So any spike generated here does not have to be delivered until after that minimum delay has expired. And so that gives you synchronization points. From exchange to exchange, you can collect all of the spikes, and then at these exchange points, transfer the spikes to where they're needed. And it's guaranteed that they won't arrive late because their delay is greater than or equal to this minimum delay. This is the typical default case that's used. So if there's an action potential in this GID, and the delay and the minimum delay are these what is 1 millisecond, then we have here that if a cell is spiking at this time, let's say at 1.475, we record that GID spike time in a buffer. And also we count the size of the buffer. When time 2 arrives, we do then the very simplest possible transfer of information using MPI all-gather at time t equals 2. And that on the target, that means that this spike gets sent to every other processor. That's what all-gather means. This buffer gets copied onto every other machine. So from a source buffer, it's a small thing, but on every machine, there's an in-host size target buffer. And then the target machine looks through the buffer that's been received by AllGather, and when it sees one that it's interested in, in its dictionary of GIDs that are sources to some synapse that it has on its machine, it puts it into a queue, a priority queue, to be delivered when time, let's say that the delay was 2. When time 2.475 comes around, it gets delivered to all of its post cells, all the synapses that may be interested in that spike on that machine. Now, if one has a lot of machines, you might say, well, why would you send spikes everywhere if not everybody wants them or needs them or is interested in them? So that alternative is, that alternative is to say, well, let's send our spikes. If we have a list of processors which are interested in any specific GID as a target for that GID, we can then, while we're computing, when this happens, during the, when this happens, we go ahead and we send the spike with MPI iSend to every processor that's interested in it. And then at the end of a synchronization interval, we just make sure that the number of sent spikes is equal to the number of received spikes, which is a much faster process than MPI AllGather. It's an MPI AllReduce. And then at that synchronization point, then the spikes that have arrived get put onto the queue for delivery. In fact, in almost all circumstances, in practical cases, the MPI AllGather is much faster than sending individual spikes. And the reason is because in most practical networks, there's a huge amount of fan out. So if a cell fires a spike, that spike probably gets sent to 1,000 or 10,000 other synapses. It's very likely that every processor then, if you have only 100 processors or even only 1,000 processors, that every one of those 1,000 processors will want that spike for at least one of their synapses. And so AllGather is much faster than 1,000 MPI sends, to a specific set of machines. Okay. And in fact, one sees that here. That is, in terms of the weak scaling, you can see that MPI AllGather, up to 64,000 processors with 2 million cells, is still 3 times faster than what one can get when there's 10K cells per connections per cell. Still much shorter in time than the MPI send receive. Okay. However, if you're trying in weak scaling to move up, if you have just a small number of 1,000 connections per cell, when you get to 64,000 cells, 64,000 processors, now only usually 1/64 of the machine is interested in those cells. And in that case, MPI send receive, above that, let me say this is AllGather. You can see that AllGather is getting longer and longer because it has to send more and more across the entire network. But even with 1,000 connections per cell, AllGather on a blue gene P is faster than iSend, iReceive. Okay. So even though only 1/64 the number of processors need a connection, it's still faster to send them everywhere with that kind of, because it's so optimized for the hardware on the computer. But if you go down to the hardware itself, and actually use the, instead of MPI, you use the hardware available lower level functions, then yes, you can make connections, so to speak, disappear from the process of transfer, basically disappear from the problem for arbitrarily large problems. Now I want to return to that issue of connectivity, which is important because it often turns out when people write network models that even though the simulation part, the run itself, scales linearly with the number of processors, it's also true that most people end up writing their code in such a way that the setup problem, the setup time, the definition of the network to actually create the network, does not scale. And the reason is because every processor generally is doing a loop over all possible cells and a loop of all possible targets, and then picking out the ones that it is interested in to actually make the connection, okay? And that, in the end, is a non-scalable thing, which is going to spell doom for the scalability of your process, no matter how large a machine you have. And so I want to introduce this parallel computational database pattern, which has to do with information exchange during setup time, to hopefully have in the back of your mind if you're developing these connectivity algorithms. So the reason I call it database is because we have to imagine that globally we have hundreds of millions of objects, hundreds of millions of cells, hundreds of millions of brain has a quadrillion synapses. Okay, and we're talking about terabyte-size data, or much larger in some of these models. And the problem then becomes, well, we have to select objects from this huge amount of data with a particular property value, right? In order to, it's a selection process. Any given number, any given processor is interested in only a trivial small amount of the total data. Okay. We call it parallel because we really need to reduce the time and space that's used according to model size divided by n-host. We add more hosts to the problem. We want to reduce the setup time. We don't want to, at least we don't want to increase the setup time. So on a BGQ, for example, that's now been decommissioned, 4 racks at 124 nodes per rack, 1,024 nodes per rack, 16 gigabytes per node, that's 64 terabytes of memory on that machine. But oftentimes for highest efficiency or highest performance, we run 32 processes per node, right, which is basically ends up a half a gigabyte that's available to any given process. So of the 64 terabytes that the model is in, any given location can only access half a gigabyte of it. Right? Terabytes, gigabytes. So there's more than 1,000-fold, 128,000-fold difference there. Why do I call it computational? Well, we have to read the data from files and distribute it among the processors. It has to be generated, analyzed, instantiated, the model's built. Okay? And communication, we don't want communication to unreasonably dominate this process. We want to really be computation limited for high performance. Also, hard disk read-write is very slow, so you never want to write data to disk and read it back from another processor, okay? Or write huge files. You don't want to write those terabyte files and then have everybody read a terabyte file and extract data. Even if while you're reading, you're extracting it, so you're not saving the. You only have half a gigabyte. Okay, so what's the pattern? Why do we need a pattern? It's because ranks have data, all together globally they have the terabyte. Ranks want the data. Of that terabyte, they're only interested in half a gigabyte of data. But no one knows where the data is, okay? Any given rank knows what it has and it knows what it wants, but it's not the same thing. Right? It doesn't know where what it wants is located on the machine, because data essentially is distributed randomly on the machine. All right. So generally speaking, all of this data is in the form of dictionaries, key data. And key is an integer or a float or a string or a tuple in Python. It's a fixed thing. And all the data is of this form. How do we find it? I know what key I'm interested, but I don't know where the key is, what machine the key is on. All ranks participate in alternating computation and communication. So here's an example. Model that was developed in collaboration with Michele is an olfactory bulb model. Mitral cells and granule cells. Mitral cells kind of spread out with their dendrites all over the bulb. Granule cells have an extremely small receptive field. Where they're located is basically where they have to get connected. The reciprocal synapse connection description is a mitral cell, its dendrite, its location on the dendrite, and then the granule cell, the granule dendrite, and the location on the granule dendrite. So basically there are all these, and then more weights and so forth, more of these tuples. Okay? One thing that's easy to do conceptually is to create all our mitral cells, and then we know precisely where they are. These are all randomly constructed cells with random locations. I don't know how many dendrites there are. I don't know how long they are. It's constructed by an algorithm, but it ends up being constructed, and one can then iterate over all the positions of the mitral cell and say, yes, I do want to construct a reciprocal synapse. And since the mitral cell, this has a particular location, I can figure out what the GID of the granule cell is that I want to actually have the connection. Okay? So I construct a mitral and all the tuples with that mitral GID. Okay? The problem is the granules don't know anything about the mitral cells. They don't know what mitral cell they're supposed to be connected to. All there are now distributed over the machine are these millions of tuples. Right? So granules don't know enough for construction of the tuples. Okay? So what we have to do to construct a granule cell with some specific G for the granule GID is we have to find all of the tuples globally throughout the machine that have this specific G. Okay? That's the problem. So we need, let's say we need several thousand out of 100 million of these things. So how do we find them? Here's so you can see in the mitral cell there's a glomerulus with a number of tufts, and then there are half a dozen mitral cells, and these mitral cells all have these dendrites that spread out all over the bulb, and there are several, there's hundreds of these tufts, hundreds of these, looking at them end on, but any individual mitral cell, is fairly sparse, and it only connects to granules which are underneath any particular location. Right? So we can easily do this for mitral cells, but any specific granule cell, which mitrals is it supposed to connect to? So it needs to find those tuples. Now, the only element that we have that can help us at MPI is something called the pi all to all. And what that means is that the source and destination are a list of in-host objects, Python objects. And basically what happens with all-to-all is that if there's, if rank 1 has a red object, a green object, and a blue object, rank 2 has a bunch of red objects, green, and so forth. Rank 3 has distributed. What we want is rank 1 wants to collect all of the red objects. Rank 2 wants to collect all of the green objects. This happens with pi all-to-all. It's basically a transpose of a matrix. Okay. So the source j on the ith rank will be copied to destination I on the jth rank. It's a transpose. But, like I say in the problem, no one knows where these things are supposed to be sent to, and no one knows where they are. Okay? So, and there's no room to have a global map for all these things. That would take more than your ½ gigabyte of available memory. So the solution, which is used ubiquitously many, many times in all of these models that want to be parallel setup and not just linear setup time with the model size, okay, is to use what's called a rendezvous rank function, in which some rank is determined by the property of a cell. Okay. Usually that is just rank equals GID percent in host. In other words, all of the GIDs would be distributed evenly now among all the hosts. In general, if you're using complex objects and you want to have a rendezvous rank, that's generally key hash, the hash of the key. Use that, and then do a percent in host, and that will then divide this hash by the in host and take the remainder. And then that's the rank that you expect the object to be sent to. All right? So basically, the idiom is that everyone sends the keys they own to the rendezvous rank. Now that doesn't mean the rendezvous rank will have more keys than the original sender, because remember, it's distributed everywhere. Okay. Everyone sends the keys they want to the rendezvous rank. The rendezvous rank sends back to the owners which ranks want which keys, and then the owners send the objects, the key value, send the value of the key to the ranks that want them. That's the most general case. This can be simplified in practice quite a bit. If you've already distributed everything according to GID, instead of balancing things, you automatically know where to send it, because if you have a granule GID, I know where it is, because it was distributed round robin fashion. Okay. If the object is very small, or not too large, I should say, then you can send the object to them. You can, rather than sending the keys, you can send the object to the rendezvous rank as well, the key, and reduce to 3 steps. And that's what's done with the problem of connecting granules to mitrals. So every program basically, from Neuron import the H. PC is parallel context. In-host is the total number of hosts, and rank then is the specific integer of this particular host, ranging from 0 to in-host -1. And we'll just assume that ngcon is a list of these tuples, and ggids is a list of the G's on this rank, the granule GIDs on this rank. So, organizing the tuples destined for rank G percent in host as a list of tuples. So basically what we say is, let's say the variable have is a list of empty lists. A list of empty lists, and the size of this have vector is in host in length. All right? So that's our destination, that's our send That's what we're going to send. All right? Now we need to fill up this vector properly. So for con in mgcon, in other words, looping over all of the tuples on this machine, not the global number, but just the ones on this machine. Con 3 is the granule cell GID, right? And so have G percent in host, that's where it's going to go. We then put con into it, that particular list of that one. And then we say, okay, just say have on the rendezvous rank now is pc.pi all to all. So that basically gives the rendezvous rank all of the G's that it needs that have that property. G percent in host is its rank. Okay, the want machine has a list of granule cell GIDs and wants all the tuples that refer to any of its G values. So we have to tell the rendezvous rank what G's it wants. It's exactly the same as before. A list of empty lists equal to, in size to the machine, number of hosts. All right? And for G and GIDs, want G percent in host, append G. Right? And then we send it. Want PC pi all to all want. So now the rendezvous rank knows who has, well, it has what the wanter wants now, because I sent the tuples. I didn't just send the keys, I sent the tuples. So I'm going to save an entire all-to-all transfer process. Okay, now the rendezvous rank, though, for simplicity, so that we're not constantly looping through its list of things, we'll make a dictionary between G and destination rank. So we look at the what came in through the want mechanism, all right? And this there are n host of them, so R goes 0, 1, 2, 3, 4, 5 up to n host minus 1. And then there is a GS list of all the G's on that particular rank. And so I basically say G to rank G create an item in the dictionary. G to rank G equals R. All the G's are distinct, but R's will have, there will be many R's for many different G's. Okay. There'll be one R for many different G's. Okay, now that I've got that dictionary, I can send back to the wanter all the tuples where those G's exist. So again, now you're starting to see the pattern, I hope. A list of empty lists. A lot of times it's maybe even more higher performance to put none in here and then create the list when it finally is non-empty. Okay, it's not a big deal. For tuples in the have list, and for all the tuples in those lists of tuples, all right, the G is T of 3. And now in the send list, it's G to rank of G, which is the proper rank, right, and append the tuple to its list. Do the all-to-all send, and now you have, the wanter has all the tuples that it wants. And it can look through those tuples and make the connections using the connect, GID connect, to make its connection to the mitral cell. These are reciprocal synapses, so there's a netcon that goes from a threshold detector on the granule to a synapse on the mitral, a inhibitory synapse, and another netcon that goes the other way exactly, which is from the threshold detector on the mitral, and there are thousands of threshold detectors on those mitral cells, to the excitatory synapse on the granule cell. Okay, so I will stop there. Thank you for your attention, and good luck in the use of this pattern.